Okay, hello and thank you for joining this Cancer Research and Oncology event. My name is Hamacher-Brady and I'm an assistant professor at the Johns Hopkins Bloomberg School of Public Health. My lab studies how mitochondria integrate stress responses with pathways like apoptosis and autophagy, and how cancer cells exploit these mechanisms to survive. In today's talk, I will focus on mitochondrial dynamics and stress signaling in cancer, and then highlight our research on the cancer testis antigen FATE1, which acts as a cancer-selective modulator of mitochondrial remodeling. So as I mentioned, my presentation has two segments. In the first, I'll give an overview of how mitochondria shape cell fate through fission, fusion, apoptosis, and autophagy. I'll connect these core processes to cancer biology, showing how tumor cells remodel mitochondrial function to support growth, evade death, and resist therapy. In the second part, I'll present our case study on FATE1, a cancer testis antigen that reprograms mitochondrial dynamics and stress responses in cancer cells. Together, these perspectives will link fundamental cell biology with emerging mechanisms of cancer cell survival. By the end of this talk, you should be able to, first, explain how mitochondrial dynamics intersect with apoptosis and autophagy in determining cell fate. Second, describe how advanced imaging methods reveal mitochondrial remodeling and stress signaling. And third, analyze how these processes are co-opted in cancer, with FATE1 as a concrete example of a tumor-selective survival factor. These objectives will frame both the basic concepts and their application to oncology. Mitochondria are central hubs for both cellular homeostasis and stress adaptation. They produce ATP, buffer calcium, and generate reactive oxygen species that can serve as signaling molecules or cause damage if uncontrolled. Beyond metabolism, mitochondria also act as gatekeepers of apoptosis and as key initiators of inflammatory signaling, for example, through reactive oxygen species release or leakage of mitochondrial DNA. These functions are often rewired in cancer, making mitochondria decisive checkpoints for whether cells adapt, die, or drive disease progression. Mitochondria comprise a dynamic organelle compartment that is constantly remodeled by fusion and fission. Fusion, mediated by mitofusins and OPA1, maintains mitochondrial function and supports survival. Fission, mediated by DRP1 and its adapters like MFF, promotes quality control and primes cells for mitophagy or apoptosis. The balance between fusion and fission is critical for determining cell fate. You can see this dynamic behavior in action in this live cell movie of neuroblastoma cells expressing a mitochondria-targeted GFP. Mitochondria are continuously undergoing fission and fusion, illustrating they are highly dynamic networks rather than static powerhouses. In cancer, this balance is often shifted towards fusion or hyperfusion, which helps tumor cells survive under stress. With this framework in mind, let's turn to how apoptosis and autophagy intersect with these dynamics. Apoptosis is the cell's built-in self-destruct program. It shapes tissues during development, removes damaged or infected cells, and maintains homeostasis. Cancer, however, often evades apoptosis. That's one of its core hallmarks. When this cell death program is muted, abnormal cells persist, accumulate mutations, and resist treatment. At the same time, this vulnerability is also what many therapies rely on. Chemotherapy and targeted agents often aim to reactivate apoptosis in tumor cells. So apoptosis matters in cancer, not only because its loss enables tumor growth, but also because restoring it remains a cornerstone of effective therapy. And a core decision point for whether apoptosis occurs sits at mitochondria, which act as the cell's gatekeepers. This slide shows why mitochondria are central to cancer therapy. When the outer mitochondrial membrane is permeabilized, referred to as mitochondrial outer membrane permeabilization or MOMP, cytochrome C is released from the mitochondrial intermembrane space to the cytosol. apoptotic caspase proteases are activated, and cell death follows. The BCL2 family sets this threshold. Anti-apoptotic proteins like BCL2, BCL-XL, and MCL1 block MOMP, while pro-apoptotic BH3-only proteins promote it by activating BAX and BUC. XIP adds a brake by inhibiting caspases. Most effective chemotherapies and many targeted agents ultimately work by pushing cells past this mitochondrial checkpoint. Tumors that overexpress anti-apoptotic BCL2 proteins can block MOMP and resist treatment, whereas BH3 mimetic drugs lower that threshold and restore apoptosis. In short, whether mitochondria commit to MOMP often determines whether therapy succeeds or fails. Autophagy, on the other hand, is the cell's recycling pathway, a way to degrade and reuse damaged or surplus components via engulfment in autophagosomes and delivery to lysosomes. When this process is directed at mitochondria, it's called mitophagy, which removes damaged mitochondria and thereby maintains quality control. In cancer, mitophagy has a dual role. Early on, it can suppress tumor initiation by eliminating defective mitochondria. But in established tumors, mitophagy often supports survival under stress, including by counteracting mitochondrial apoptosis signaling. In this way, mitophagy links mitochondrial quality control directly to cancer progression and therapy resistance. Having outlined how mitochondria intersect with apoptosis and mitophagy, let's now turn to the tools that allow us to investigate these processes. Much of what we know about mitochondrial biology and signaling has come from fluorescence microscopy. Using mitochondria-targeted fluorescent proteins, dyes, and probes, we can visualize the organelles dynamics inside the cell. High and super resolution microscopy now let us resolve mitochondria with remarkable detail, revealing mitochondrial morphology, organelle contacts, MOMP, and even individual mitophagy events. Live cell imaging adds another layer by capturing not only spatial but also temporal and microenvironmental cues, such as changes in pH in the lysosome or such as calcium, as cells respond to stress. This power to combine structural, dynamic, and functional readouts is what has made fluorescence microscopy indispensable for understanding how mitochondria integrate stress signaling. Here are examples from our own work where we used high-resolution imaging to visualize mitophagy. On the left, you see a mitochondrion being engulfed by a forming autophagosome to be delivered to lysosomes for whole organelle degradation. By closely examining mitochondrial and endolysosomal organelle compartments during stress, we discovered that mitochondria can also directly be targeted by endolysosomes. In the middle of the slide, you see an instance wherein apoptotic mitochondria have actually internalized RAB7 positive endolysosomes. We determined that these direct endolysosome mitochondrial interactions are mediated by ubiquitination events and can carry out selective degradation of mitochondrial subdomains, removing only portions of the organelle while leaving the rest intact. You see this on the right side of the slide. And this demonstrates that mitochondria can be remodeled in a highly localized and selective manner, a finding that would have been invisible without advanced imaging. Beyond mitophagy, using live cell imaging, we uncovered a completely new layer of apoptosis regulation, mediated by dynamic interactions between endolysosomal vesicles and mitochondria, which you can see in the movie that I'm playing here, showing a single mitochondrion being targeted by endolysosomal vesicles during apoptosis, supporting BAX-mediated MOMP. This discovery highlights how spatiotemporal resolution can reveal unexpected crosstalk in mitochondrial stress signaling. Stepping back from these imaging examples, what do they tell us about the bigger picture? Mitochondrial dynamics directly shape autophagy and apoptosis. Fragmentation primes mitochondria, for clearance by mitophagy and for apoptotic permeabilization. Elongation helps mitochondria resist degradation and sustain metabolism. Hyperfusion often emerges under stress as an adaptive survival response. This close integration of mitochondrial dynamics, autophagy, and apoptosis underscores the central theme: mitochondrial morphology is not just a bystander, but an active regulator of how cells respond to stress. This provides the conceptual bridge to our case study on FATE1, a cancer-selective survival factor that manipulates this very axis. With this, we are now moving into the second part of the presentation, which focuses on our case study of FATE1, which is short for Fetal and Adult Testis Expressed 1. FATE1 is emerging as a fascinating example of how tumors can exploit mitochondrial dynamics to resist apoptosis. In the following slides, I'll walk you through how FATE1 compares to its homologue, the mitochondrial fission factor MFF, what makes it distinct, and how these features define its novel role as a cancer-selective regulator of mitochondrial dynamics. FATE1 is a cancer testis antigen, meaning its expression is normally restricted to testis, but becomes aberrantly expressed in many cancers, including in adrenal, colorectal, breast, and Ewing sarcoma. It was first identified in small cell lung cancer as a factor that protects cancer cells against chemotherapy and has been proposed to suppress apoptosis by degrading BH3-only proteins or by uncoupling ER mitochondria calcium signaling. What was less clear until recently was whether FATE1 might also regulate mitochondrial morphology itself, given its sequence similarity to MFF, a mitochondrial fission factor. To address this, we performed sequence analyses comparing FATE1 and MFF, which promotes mitochondrial fragmentation by recruiting DRP1. Both proteins share a C-terminal coiled coil and a transmembrane domain, but there's a critical difference. MFF contains two conserved N-terminal repeat motifs, R1 and R2, which are essential for recruiting DRP1 and for initiating fission. FATE1, in contrast, lacks the R1 motif and only has a partial resemblance to the R2 motif. The structural divergence immediately suggested to us that FATE1 may not function as a canonical DRP1 receptor and instead might play a distinct role in shaping mitochondrial architecture. Using wide-field fluorescence microscopy, we compared the subcellular localization of GFP-fused FATE1 and of GFP-fused MFF in MCF7 breast cancer and HeLa cervical cancer cells, which are shown here. We found that, as has been reported by others, FATE1 localizes to both mitochondria and the endoplasmic reticulum, or the ER, unlike MFF, which is strictly mitochondrial localized. You can see that FATE1 decorates mitochondria, but also concentrates at perinuclear regions and can even induce ER clustering. This dual localization, especially at perinuclear ER clusters, suggests that FATE1 might act directly at mitochondria ER contact sites, further emphasizing that its functions diverge from its homologue MFF. Using STED super-resolution microscopy, we were able to resolve FATE1's distribution on mitochondria with much higher precision. These images show that FATE1 localizes specifically to the outer mitochondrial membrane, where it decorates the organelle surface in a punctate pattern. Co-staining with mitochondrial markers confirmed that FATE1 resides at the outer membrane, positioned exactly where it can act. Given its localization to the outer mitochondrial membrane and homology to MFF, we asked whether FATE1 could act as a DRP1 receptor. But across co-immunoprecipitation, subcellular fractionation, and high-resolution imaging, the answer was consistent. FATE1 does not bind DRP1, nor does it recruit DRP1 to mitochondria. In contrast, MFF, as expected, robustly brings DRP1 to the outer membrane and drives fragmentation. Therefore, despite their structural similarities, FATE1, lacks the R1 motif required for DRP1 engagement, and diverges mechanistically from MFF. So if FAT1 is not a DRP1 receptor, does it still affect mitochondrial morphology? To test this, we performed stable knockdowns in HeLa cells. Depletion of FAT1 shifted the population toward fragmented mitochondria, reducing the fraction of cells with elongated or interconnected networks. This loss of function phenotype suggests that endogenous FAT1 normally acts to preserve mitochondrial network integrity. Without it, cancer cells become more prone to fragmentation, a state typically associated with stress and apoptosis sensitivity. Conversely, overexpressing FAT1 produced the opposite phenotype. Mitochondria clustered and elongated, and many cells displayed hyperfused networks spanning the entire perinuclear region. The extent of hyperfusion correlated with FAT1 expression levels. Not shown here, but even endogenous upregulation of FAT1 by the transcription factor SF1 was sufficient to drive this phenotype. Together, loss and gain of function studies demonstrate that FAT1 antagonizes fission and actively promotes mitochondrial elongation. To understand how FAT1 localizes and functions, we generated a panel of deletion mutants. The transmembrane domain was required for membrane insertion, while the coiled coil domain was essential for mitochondrial targeting. Without these domains, mutants mislocalized to the cytosol or the ER and lost the ability to promote hyperfusion. Interestingly, N-terminal truncations increased mitochondrial targeting, suggesting that the N-terminus normally inhibits outer membrane insertion. Overall, mitochondrial localization was the key determinant of FAT1's ability to remodel mitochondrial networks. We then asked whether FAT1's effect on mitochondrial morphology depended on the fusion machinery. Co-immunoprecipitation experiments in HeLa cells showed that FAT1 specifically interacts with mitofusin 2, but not mitofusin 1. You see this on the left side of this slide. On the right side, you will see that in mouse embryonic fibroblasts lacking both mitofusins, so mitofusin 1 and 2 double knockout cells, FAT1 could not induce hyperfusion. Reintroducing mitofusin 2 fully rescued this phenotype, whereas mitofusin 1 did not. These results established that FAT1's profusion activity is mediated by mitofusin 2, identifying it as a novel effector of FAT1-mediated mitochondrial hyperfusion. Building on the mitofusin 2 dependence, we asked whether FAT1 also buffers the fission machinery under stress. When we challenged cells with TNF-alpha or interferon gamma, or with the mitochondrial uncoupler valinomycin, GFP control cells rapidly accumulated DRP1 puncta on mitochondria and began to fragment. In contrast, GFP FAT1 cells showed markedly reduced DRP1 relocation across all conditions, and mitochondrial networks remained elongated. You can see this in the representative images on the left and in the quantification on the right. Both the percentage of cells with DRP1 puncta and the puncta burden per cell are significantly lower with FAT1 overexpression. In short, FAT1 suppresses stress-induced DRP1 assembly at the outer mitochondrial membrane. We then scored mitochondrial morphology under the same stresses. In GFP control cells, TNF-alpha, interferon gamma, and valinomycin drive a strong shift toward fragmented mitochondria. In GFP FAT1 cells, networks remain continuous and often tubular or hyperfused. The representative images on the left and the population scoring in the middle or toward the right tell the same story. FAT1 robustly protects mitochondrial architecture across distinct stressors. Functionally, this positions FAT1 as a buffer that antagonizes the DRP1-dependent fission program and sustains a profusion state during inflammatory or mitochondrial insults. an ability that likely enhances cancer cell survival under therapy-induced stress. Summarized in the schematic here to the right. Bringing these findings together, here's a model to illustrate how Fate1 diverges from its homologue MFF. MFF recruits DRP1 through its R1 and R2 motives, leading to mitochondrial fission and priming cells for apoptosis. Fate1, which lacks R1, instead engages mitofusin 2 to promote mitochondrial hyperfusion. Together, these mechanisms establish Fate1 as a cancer-selective survival factor that remodels mitochondrial networks to withstand stress. Of course, these findings are not just mechanistic details. They also carry broader implications for cancer biology and therapy. Cancer testis antigens like Fate1 are compelling therapeutic targets because their expression is normally restricted to immune-privileged tissues. Elevated Fate1 levels correlate with poor prognosis in cancers such as adrenocortical carcinoma, colorectal cancer, and breast cancer. Mechanistically, previous studies have proposed additional ways by which Fate1 suppresses apoptosis, either by promoting degradation of BH3-only proteins, as shown by the Whitehurst lab, or by uncoupling ER mitochondria calcium transfer, as reported by the Lally lab. Our data add a third dimension, showing that Fate1 enforces mitochondrial hyperfusion through mitofusin 2. Together, these complementary findings highlight Fate1 as a cancer-selective survival factor that tumors may exploit through multiple, possibly interconnected, pathways. Disrupting these functions, whether the Fate1 mitofusin 2 interaction, its impact on ER mitochondria coupling, or its effects on BH3 proteins, could provide new therapeutic opportunities, while its restricted expression profile makes it an attractive immunotherapy target as well. To wrap up part 2, let me highlight the main take-home messages in where we see this research is heading. First, Fate1 does not function like its homologue MFF. It lacks DRP1 receptor function. Second, Fate1 promotes mitochondrial hyperfusion through mitofusin 2, preserving network integrity under stress. Third, we propose that this activity provides a survival advantage to cancer cells, making Fate1 a cancer-selective regulator of mitochondrial dynamics and a potential therapeutic target. Looking forward, we are investigating how Fate1 localization is regulated, whether post-translational modifications influence its distribution between mitochondria and ER, and whether disrupting the Fate1 mitofusin 2 interface can sensitize cancer cells to therapy. Given its restricted expression profile in healthy tissues and its central role in cancer cell stress resistance, Fate1 is not only a fascinating basic biology discovery, but also a promising target for future therapeutic strategies. To close, let me return to the learning objectives we began with. So we've seen how mitochondrial dynamics intersect with apoptosis and autophagy to determine cell fate. We've also explored how advanced live cell and super-resolution microscopy reveal the spatial and temporal coordination of these processes. And we've analyzed how the cancer testis antigen Fate1 reprograms mitochondrial dynamics, linking its unique activities to cancer cell survival and therapy resistance. With this, I'm at the end of my presentation and would like to sincerely thank my lab members, collaborators, and funding agencies who made this work possible. And thank you for your attention today. I hope I've shown you how mitochondrial remodeling is central to stress adaptation and cancer survival, and how factors like Fate1 reveal cancer-selective mechanisms that we may be able to target. Feel free to reach out by email or connect with me on LinkedIn if you'd like to discuss or follow up. Thank you.